So the next topic we're going to discuss is human genetic variation. An important concept to know is that we are mostly identical in our genetic makeup. In fact, for every 1,000 nucleotides, there will be only a difference of a single nucleotide between any two individuals. This was a big surprise whenever they did the Human Genome Project. They thought that they would be able to find a lot more differences between us. Realize that allele means any variation at a specific genomic locus. So if you have two different alleles, you have two different versions of the same gene. If a version is present in less than 1% of the population, people generally refer to it as a mutation. So one nucleotide mutation that just replaces it with a different nucleotide, if that specific allele and nucleotide sequence is present in less than 1% of the population, it is a mutation. If it is present in more than 1% of the population, it is called a polymorphism. And if it is a single point mutation that's present in more than 1% of the population, it is a SNP or SNP, single nucleotide polymorphism. And we will discuss that more. So genetic variation is important for survival, right? So we get variation through recombination and mutation. Recombination really refers to our sexual reproduction. It refers to allelic shuffling, homologous recombination, and if any of those terms are really unfamiliar, just click on the links and you should be able to review them. Mutations are another factor that really drives variation, and it creates different versions or alleles of the same gene. Remember, we have 23 pairs of homologous chromosomes. We get one from mom, one from dad. Right, and so you see here, this is representing a different DNA sequence in the same gene, so you have different alleles. We also have independent assortment, right? So genes are inherited independently of one another, and then you will also get this reassortment of all of these alleles. You'll get one from a mom and one from a dad. This is showing that during meiosis, it splits into different sperm. On top of that, we create additional variation by homologous recombination. And so as they duplicate, the chromosomes duplicate, now you have chromatids, and they line up together, they will swap out different genes. And so not only will it be random, 1, 2, 3, 4, which one of those chromatids that you inherit, the chromatids won't even be identical in nucleotide sequences as they were in your mom or dad's cells because of this recombination. So that gives you even more variation. And then, of course, the independent assortment. So let's talk about mutations, because this is really the very important part that we will focus on in this class. So mutation is just a process that changes the DNA sequence. It's not good or bad. The impact of a mutation on gene expression or otherwise will depend on a few different things, right? So you can have inherited mutations, and those mutations occur in sex cells that can be passed off to the offspring. You can also have spontaneous or acquired mutations, and acquired is also divided into spontaneous, and this is just what happens during the DNA copying process that occurs prior to cell division. So in your in the cells in your body. Now those won't necessarily be passed on to the next generation. You can also have induced mutations, and again, these won't necessarily be passed on to the next generation. They have to occur in your gametes to be passed on. And then the effect of the mutation will depend on the type of mutation. So if it's a point mutation, that single base change in a DNA sequence, it may do nothing, like it may be inconsequential. But if it changes the amino acid that's coded, then the mutation is termed a missense mutation. And that point mutation or missense mutation is something that occurs in sickle cell. You can also have deletions or insertions or translocations. And so what this is saying is that you can have a certain gene, a particular gene that codes for this particular protein, and the type of mutation might make it a different shape. It might make more protein, or it might make zero protein. And the outcome of that in terms of your trait or disease status is going to be very different, right? Now, that is if a mutation occurs in the gene, the protein coding part of the DNA sequence. But remember, 99% of our DNA does not code for a protein. And so if it occurs in a different location, then the effects might be different. So this is just showing the different types of mutations. So single nucleotide polymorphism, or SNPs. Whenever you look at the media and you're looking at different articles, a lot of times they're talking about SNPs, and that means a single point mutation, a single base pair difference, right, in an allele. And remember, polymorphism implies that this particular allele is present in more than 1% of the population. So it has to have two different versions that are present in more than 1% of the population. So you can have a SNP in the coding region of the DNA, which might impact the protein, right? It might impact the shape if it impacts the amino acid sequence. You can have it in one of the regulatory sequences in the non-coding part of the DNA. And this might change the amount of protein produced. So if you have those on and off switches, like we talked about before, say this mutation impairs your ability to turn the gene off, and so you just have more and more and more expression, then you're going to have a lot more protein produced. Now, it also could be outside of the gene sequence, and it might not have any effect on the protein production or function. And this is what usually happens. So the majority of SNPs, to our understanding right now, are in places where it really doesn't matter. So SNPs are very, very common. Greater than 10 million SNPs are present in humans, and the average person has about 3 million of these. But again, most of these are located in areas where they are inconsequential. So another example of the different outcome, or how a mutation can create variation in gene expression, is if that mutation, and we're talking about these SNPs again, are in these regulatory sequence, these switches, on and off switches. And so I want you to watch the video. It's in the Blackboard module, and it gives an example of two different populations on different parts of the world who have this phenotype of lactase persistence. So they can drink milk as adults. Most people in the world cannot drink milk as adults. It is supposed to turn off when you are about 4 or so. And I want you to watch that and see what type of mutation creates this phenotype or this trait. I also want you to take away that some mutations can be good, right? And then So how common are these mutations in switches? Very common. So they're much less studied. Most of the genetic, mutation research is looking at these protein coding regions. So we are just finding out more and more about variations in switches or even switches themselves. And again, they work in a very complex way. So you can have one switch that turns on numerous genes, and you can have numerous switches that turn on one gene. And they are not necessarily close or linked in the same chromosome, actually. And so one relatively recent study just looked at all the variants that they knew about in a particular DNA study, and then they tried to identify all of the different switches or the variations of switches in a bunch of people, basically. That's as simple as I can explain it. And you see that, like, left ventricle, there's like almost 4,000 different variations in these switches. And so our understanding of how DNA expression is influenced by these switches are just is really in the beginning stages. Okay, go ahead and finish the module, and after you review the single gene disorders, I want you to pay particular attention to sickle cell anemia because that's what we will discuss in class.